So thank you, Matteo, and thank you, organizers, for this interesting day. Intracellular organelles do not act as independent entities, but they exchange signals and metabolites in order to maintain cellular homeostasis and answer to cellular stimuli. And they do this at membrane contact sites, which are specialized spatial and functional hubs in which the membrane of two organelles are really close to each other without fusing. And they are involved in a lot of different cellular processes, as you can see from this image. And more interestingly, their dysregulation has been associated to different pathologies. So from here, the need to study such contacts. But how can we study them and how can we study their dynamics? So electron microscopy does not allow the visualization of living cells. Confocal microscopy does not have enough resolution, while split fluorescent protein, the majority of them, they are endowed with poor reversibility. So in 2019, this group of chemists developed this split-fast system that is a reversible complementation system that has been developed for the visualization of protein-protein interaction at first. And this system is based on the fast protein, which is not a fluorescent protein per se. And this protein has been divided in two portions, the N-fast and the C-fast. Only when the two portions are really close to each other, they can bind and reconstitute a pocket which can bind a fluorogen, which is not fluorescent, but becomes fluorescent when it binds the reconstituted fast protein. You can use different fluorogens, so you can have spectral flexibility, with the exception of the far red fluorogen that you can see here, that is the only one that is able to bind to far red and fast. I mean, only the N-fast portion, is able to bind the far red fluorogen, while the N-fast can bind also the green. So we like this system, and so we targeted the system to organelles. You can see here the example of ER and mitochondria. And so you can visualize fluorescence only at the contact site. And to visualize it, you only have to add the fluorogen under the microscope, and you can see that just after 10 seconds, they are enough to visualize the full complementation. This is what you see at the microscope. So you see the signal coming from the reporter that, as you can appreciate, is actually present where the two organelles co-localize. This system, as I told you, is fully reversible because if we target a portion of the protein to the mitochondria, and the other portion to the cytoplasm, and then we perform permeabilization of the cell, the cytoplasmic portion is really free to exit the cell, and so we have a prompt decrease in fluorescence. We like the system, and we generated two versions of the probe, a short and a long version that is able to visualize shorter and longer contacts between the organelles. And we generated inducible stable clones expressing the two versions. And in order to quantify these contacts, we developed a Python-based script that is able to perform a mask on the mitochondrial signal and on the signal of the contact site. And then it calculates the percentage of the mitochondrial surface that is in contact with the ER. With this quantification, we got that around 10% of the mitochondria is in close contact with the ER, while the 20% is in long contact with the ER. With these clones, we studied the dynamic modulation of contact site, and the reversibility of the probe allowed us to see changes in increase and decrease of the contact site, and allowed us to understand that the increase in contact site between ER and mitochondria that is normally reported for ER stress is only transient. In fact, we saw an initial increase, but then followed by a decrease of such contact. contacts. Moreover, if we washed away the tunicamycin stressor, we could restore the physiological extent of contact site. We got an increase in contacts also upon starvation, an increase in cell density, or a decrease by blocking the cell cycle. We also perform CLEM, which is correlative light electron microscopy, and this further validated our reporter, since, as you can appreciate, the fluorescence comes actually at the site where the mitochondria and the ER are really close. And we also quantified the contact site by TEM in transfected and untransfected cells without seeing any difference, meaning that the reversible nature of the reporter does not force the physiological extent of the contact site. We studied this system also to visualize and study molecular mechanisms. We started from the SOSI, which is a mechanism that occurs when the ER calcium is depleted. So upon ER calcium depletion, we have the STIM protein that changes conformation. Then it approaches the plasma membrane, there binds the ORA channel, permitting the entrance of the calcium from the plasma membrane, and then the refilling of the ER through the Cerca pump. By targeting a portion of the probe to ER and the other to the plasma membrane, we saw the increase in contact site upon ER calcium depletion, but we also targeted the STIM1 to the one portion to STIM1 and one portion to the plasma membrane, and we were able to see that STIM1 approaching the plasma membrane occurs before the increase in ER plasma membrane contacts. And so we use this system to visualize mechanisms also at ER mitochondria contact site. And my colleague, Dr. Paloma Garcia Casas, that worked with me in this project, she demonstrated that upon ER calcium depletion, you not only have an increase in contacts between ER and plasma membrane, but also between ER and the mitochondria. And that this increase is transient, because when you have a refilling of the stores, you can get back to the physiological extent of the contacts. She also validated this system in vivo using C. elegans, expressing the probe and treated with tapsigargin, that is an inhibitor of the Cerca pump of the ER. And she also found that STIM1 and his activator TMEM110 are molecules involved in these mechanisms. We tried also our sensor in pathological conditions using lentiviral infection. We expressed the reporter, and we got an increase in contact site in all the samples from Alzheimer's disease, in particular familial Alzheimer's disease, both in astrocytes, neurons, and human fibroblasts. Taking advantage of the spectral flexibility of the reporter, you can also visualize two different types of contact sites in the very same cell. In fact, if you tag the N-fast to the mitochondria and the far red N-fast to the plasma membrane, that we remember the N-fast is the only one far red that can bind the far red fluorogen, but it can also bind the green fluorogen, and then you can also tag the ER by the C-fast. Doing that, you will have the contacts between ER and mitochondria, which are the only contacts that are only green, and the other that are both green and far red. So you can distinguish the two also only by that, or you can more elegantly separate the fluorescence coming from the green in the two different types of contacts by FLIM. That is what actually we did. And using the same approach, we also saw in the very same cell short and long contacts, and we saw that the majority of the majority of the short contacts are next to a longer one, and the majority of longer one are peripheral to short ones. And we also saw that in tomography from untransfected cells. But the other great advantage of the reporter is the dynamicity. So we used this probe to visualize the dynamics of single contact site. And as you can see here, we were able to see the transient docking of the ER to the mitochondria by the appearance and disappearance of the fluorescent signals. So we tracked these contacts, and we saw that they are able to undergo fission and fusion events in very simple trajectories, but also more complicated ones, as the one that I reported here. However, if by quantification of these the fission and fusion events of each tracks, we saw that their number was very similar, meaning that under physiological conditions, these contacts are very dynamic, but their number remains quite constant. Finally, we segmented the ER, and we noticed that the contact site involving the tubules are less in number respect to the contact involving the sheets of the ER, and by tracking the single contacts, we also saw that these contacts, the one with the tubules, are also more labile. In fact, they last only 100 seconds in mean, respect to 150, that is the mean of the contacts with the sheets. Last but not least, we also decorated our reporter with calcium sensing domain, and we named this probe Princess, from probe for interorganel calcium exchange sites based on split fast. And as you can appreciate, we got an increase in fluorescence only at the contact site. You cannot see very well here, but at the beginning, you already have some dots, which are the physiological contact site. And then upon the calcium release by histamine from the ER to the mitochondria, at this contact site, you register a calcium hotspot. This calcium hotspot is registered by the reporter, and so you have an increase in fluorescence that, as you can see, is really transient. So, concluding, our new probe has a very high spectral flexibility, reversibility, and adaptability to different experimental conditions. They follow the dynamics of contact site, and also they can measure the associated calcium signaling if you use the Princess. They are suitable for in vivo studies, and this reporter permitted us to see a new mechanism that involves STIM1 in the transient increase in ER mitochondria contact site upon ER calcium depletion. I would like to thank my former laboratory, Professor Paola Pizzo, and especially my supervisors for the PhD, Professor Paola Pizzo and Dr. Riccardo Filadi, which also have been the mentors for my first postdoc. And then I would like to thank my current laboratory, the Professor of Luigi Leanza in the Department of Biology, and especially the professor who gave me the opportunity to start the study of contact site in the field of tumorigenesis. And of course, all our collaborators, findings, and all you for the attention.